decades, scientists assumed mice were essentially colorblind, that color vision was a primate luxury. Turns out that's wrong. And understanding how mice see color is opening new doors in both neuroscience and biomedical technology. Hi, I'm Stefania, a Marie Curie doctoral research at Institut de la Vision in Paris, part of the Be Light network and community. My work lives at the crossroads of light, color, neural computation, and machine learning. In our lab, we record from hundreds of retinal ganglion cells simultaneously using multi-electrode arrays, and then we build probabilistic models based on Gaussian processes that learn how its neuron transforms a natural color scene into a pattern of electrical spikes. What makes this exciting and interesting is that the model doesn't just fit the data, it reveals which features of the visual world each cell actually cares about. Some cells encode brightness, for example. Others are tuned to chromatic contrast between ultraviolet and green light, the two channels of mouse color vision. Understanding these coding strategies matters far beyond the mouse. It informs how we think about vision restoration, how we design optogenetic therapies, and how we build bio-inspired algorithms for medical imaging. The retina is the brain's first interpretation of light, and by decoding its language, we're learning to speak it.
Did you know that you can interact with digital world through your gaze? What it seems to be impossible nowadays, it's reality, thanks to eye tracking technology. Eye tracking technology has a lot of application. For example, it can help us with the diagnosis of impairment of visual disorders and neurological disorders. My group and I at IMO in Barcelona are trying to build an eye tracking system integrating deep learning algorithms to early detect visual impairments in group of children. Our goal is simple: make vision clearer, accessible and easy, because everyone deserves a vision clear of the world.
Our vision is dependent to retina. Retina is the back part of our eyes, and it consists of many cells that captures what you see into your brain. Usually doctors use normal cameras to inspect this area, but for seeing all the details you need more than this. We are developing multispectral cameras that will be integrated with AI to not also show the doctors and ophthalmologists a better look, but to give them some predictions and help them to diagnose better diseases and biomarkers.
Hello, my name is Raul Urriza and I'm a PhD student at the Department of Biophysics at the Nicolas Copernicus University in Toruń, Poland. As a researcher in the P-Light, my project is devoted to a new generation of ophthalmic diagnostics that includes information on eye biomechanics. I'm going to develop a new methodology to probe the response of the tissue to external mechanical stimulus in a non-invasive way. By rapidly scanning the light beam over the sample, we will acquire such displacement information over a large volume, and I will incorporate this approach into a high-resolution microscopy setup, allowing in vivo mapping of the biomechanical properties of ocular structures such as the cornea, lens, and sclera. Additionally, I'm going to develop mathematical models and dedicated machine learning algorithms to improve the clinical utility of this device. What is the expected outcome? Well, we do believe that the result of my project will enable new insights into the relationship of ocular biomechanics and pathological conditions such as keratoconus, which will pave the way for next generation ocular diagnostics.
Imaging a future where deadly heart rhythm are stopped not with the painful shock but with the flash of light. I am Khazar Abbas and our research group turned that vision into the reality. We are developing a real-time feedback system using optogenetic, a technique where light-sensitive ions are embedded into the cell membrane. With advanced imaging and a smart algorithm, we are monitoring arrhythmia, and when they happen, we are guided light pulses to terminate it. This means fewer side effect, and lower energy defibrillation method. Our work with biomedical research groups bring physics, bioengineering, and cardiology together. We are offering not just innovation but hope. Thank you.
Every heartbeat tells a story about our health. When something's wrong with our heart, it often shows in electrocardiograms, and these are the electrical signals the doctor uses to see how our heart's going. But what if we could read these signals before troubles begin? So that's our research to detect early signs of dangerous rhythms that could be lead to strokes or heart attacks. Because these signs are very subtle and often invisible to the naked eye. And our goal is to make heart monitoring more predictive so we can understand and react before it's too late.
Imagine a world where your health monitoring is as simple as looking into a screen. No wires, no needles, no discomfort. Just a quick scan. This is now possible with the help of remote plethysmography, RPPG, a non-invasive health monitoring procedure. We are developing multimodal AI algorithms that extract vital signs and biomarkers from thermal and optical facial images. By capturing subtle variation in skin texture, blood flow, and temperature, our AI model can monitor critical health indicators in real time, all without any physical contact. This technology promises a proactive and seamless health management. It's about providing accessible and continuous health monitoring either from your home or a clinic without ever touching you. AI powered facial skin analysis is non invasive, accessible, and personalized. The future of non invasive health monitoring is here.
Patients suffering from atherosclerosis develop plaques in their arteries. Vulnerable plaques are those that are at risk of rupturing, causing potential blockages in the brain, resulting in stroke. About 20% of strokes are a result of atherosclerosis, so there is a strong need to develop methods of identifying at-risk plaques before they rupture. In the Functional and Molecular Imaging Lab at UZH and ETH Zurich, we are aiming to solve this problem using optoacoustic imaging. Optoacoustic imaging is similar to ultrasound in that it uses sound to generate images, but in this case, it uses laser light to generate ultrasound waves inside the human body before exterior detection. A key advantage of this method is that it can measure very specific types of molecules. And so, for example, it is known that plaques with high fat content tend to be more vulnerable. So by combining optoacoustic imaging with machine learning models, we may be able to predict a patient's risk of stroke earlier than currently possible.
Hello everyone. Picture the world from Earth's orbit during the rainstorm, with all of the people carrying their umbrellas. Can you pinpoint their exact position? A similar challenge is faced by microscopy nowadays. Due to light's wave nature, two molecules on a distance as close as a COVID particle size appear as one fuzzy spot. But in 2014, three brilliant scientists won a Nobel Prize for breakthrough. By observing only a few molecules on each image and collecting their positions individually, they achieved resolution 10 times better. But the question stands, can we go even further? Scientists nowadays are exploring molecular orientation. Just as knowing in which hand someone holds an umbrella reveals their exact position, molecular orientation allows us to sharpen the image and get even better resolution. Computational models suggest that this should work, but will it succeed in practice? Only the time will tell.
Scientists have long been able to look inside the cells, but not deep enough to see how individual protein molecules interact. Our imaging technology changes that. We physically expand the brain and use the computational precision method to look down at 1 nanometer resolution scale, which is 200 times better than the standard microscope. Our one microscopy allows us to study protein molecules at the molecular level inside the brain, which is offering insight into the memory and learning, the root cause of Alzheimer and autism. It is not just better imaging, It is imaging at one molecule at a time.
Imagine diagnosing a rare disease from a single cell image. At the confocal microscopy and cellular imaging unit of Hospital San Juan de Déu in Barcelona, advanced microscopy meets artificial intelligence to change how we detect and understand rare diseases like muscular dystrophies and neurogenetic disorders. Using super resolution imaging, we dive into the hidden structure of human cells and tissues, uncovering details that traditional techniques can't see. And thanks to machine learning and computer vision, we curate and decode these complex images, extracting key biomarkers and discovering new ones that make diagnosis easier, earlier, and more accurate. Because this is the future of medical imaging, and this is how we make the rare diseases a little less rare.